Hello everyone, again, welcome to MMBS-111 Introductory Microbiology. This mini lecture will discuss the external structures of the bacteria and archaea. This is mini lecture 3.2 and covers the content within section 3.2 of chapter 3. The learning outcomes for this section of the chapter and this mini lecture are first to name two appendages and their function, define chemotaxis, explain what the S layer is and its function, explain what the glycocalyx is, and name at least two of its functions. So we're going to start with appendages, the very external structures of bacteria. And appendages have two primary functions. Either they are used for motion or motility, such as flagella and axial filaments, or they help the organism attach to its location or another organism. And that is the function of the fimbriae and the pili. So the image that you see here is the basic structure of a bacterial flagellum. Its primary function is movement, and it has three distinct parts included in the structure. The first part is the filament, and this is the part here that extends out away from the cell and creates the motion. The hook you see here is where the filament attaches to the basal body, and that's the structure here that's composed of several rings and a rod. And the motion of the hook with these rings and rod creates this sort of circular action, and that's what creates the movement in the flagella and allows the organism to move in its environment. There are a number of arrangements of flagella depending on the organism, and we're going to look at each one. The first that we're going to talk about is a polar arrangement, and that means that the flagella is attached to an end of the organism. So first we have monotrichous. Mono means one. So a monotrichous arrangement means that the organism has a single flagella attached at one end of the cell. Then we also have lophotrichous. That's this arrangement here, and this just means that there are multiple flagella attached at the same point. And finally, we have amphitrichous, and this means that there are flagella attached at both ends or poles of the cell. And it could be single or there could be many attached at each end. Finally, we have paratrichous. And a paratrichous arrangement means that there are flagella attached all over in random points on the surface of the cell. And that's what you see here. Flagella are, as we discussed, used for movement, and we're going to talk briefly here about chemotaxis. Chemotaxis is the movement of bacteria in response to chemical signals, and those chemicals are chemicals that are present within the environment. Positive chemotaxis means that the organism is moving towards a favorable chemical stimuli. This could be a nutrient that is detected within the environment that the organism is moving toward. Negative chemotaxis is the opposite. This chemical is causing the organism to move away from it. So it acts as a repellent, and that could be any other poisons or unfavorable chemicals that are detected within the organism's environment. Flagella also act in two ways to create motion. We have a run, which is the flagella is moving or rotating in a counterclockwise direction, and this causes the organism to move in a smooth, sort of linear direction. A tumble occurs when the organism stops and changes direction. So this causes the flagella to stop rotating, and then the organism will change its course, and that sort of run will occur again. So here are some images of what that might look like. So we have a tumble here where the microbe has changed direction, so it has stopped. It's sort of rotating its direction, and then we have this clockwise. It's showing it should be counterclockwise motion of the flagella, so the organism is going to move in that run. And then we have some images here of what chemotaxis might look like in the motion of an organism. So if we had no attractant, there was no chemical stimulus or repellent detected within the environment, then there would sort of be this random motion of the organism. We wouldn't see any long movements or distinct changes in direction, really. Here, when we have an attractant, we see that there's definitely a direction that the organism is moving. Okay. So another type of flagella is the periplasmic flagella, and these are only found within the spirochetes, and they have a very unique structure. So the spirochetes are the corkscrew-shaped bacteria. They have this helical shape, and they have this outer membrane that you can see right here. The flagella is formed within the cytoplasmic membrane or the cell membrane, but it doesn't pass through the outer membrane. So it remains enclosed within the outer membrane. It is also attached at both ends of the cell. So you can see it sort of wraps itself around the protoplasmic cylinder here. And when these flagella move, instead of getting a run and tumble like action that you see with the other flagella, it is a very wriggly, shaky sort of motion. And that's due to the location of their flagella. These are also known as axial filaments. Okay, so axial filament, periplasmic flagella, we're talking about the same structure. All right. So let's talk about the other appendages. So we have fimbriae. You can see here in this image, it's sort of the fuzzy hair-like structures on the outside. Okay, they're small, kind of like the bristles on a hairbrush, and they allow an organism to adhere tightly to its surface, whether it's host tissues or the surface of an implant. And it also allows bacteria to adhere to each other. These structures are very important when we start talking about biofilms as well. The pili, or a pilus, you can see right here, these structures allow one organism to share genetic material with another organism. This only happens between an organism that possesses pili, and typically these are always gram-negative organisms. Okay, although they have been found in some gram-positive organisms, these are primarily a gram-negative structure. And this allows reproduction or conjugation, the passing of DNA from one organism to another. This is generally how genetic instructions for antibiotic resistance are passed from one gram-negative bacterium to another. All right, so let's talk about the external layers of bacterium. So first we are going to talk about the S layer. You can see right here, and the S layer is just outside the cell wall. It is single layers of proteins, and these are thousands of copies of a single protein that are linked together and form this very protective layer, kind of like chainmail. S layers are produced in organisms that inhabit particularly hostile environments where temperature is high, or there's a very acidic pH, where the organism needs additional protection from its environment. The glycocalyx, you can see right here, is the very outer layer, and it is a coating of repeating polysaccharides or glycoproteins, depending on the organism, and it can produce one of two structures. Either a slime layer, which is a very loose layer that surrounds the outside of the cell and protects it against water loss and loss of nutrients, or it can produce a capsule, and a capsule is much more tightly bound. It is denser. It's got the consistency of nutrient agar, and the capsule is also protective, but it also is useful in biofilms and increases an organism's ability to cause disease. And you can see an example here. Here's the cell itself, and then this layer right here is the capsule. Okay. All right, so let's talk about functions here, specialized functions of the glycocalyx. You can see the images here that talk about biofilms. So let's talk about capsules first. A capsule is formed by a number of pathogenic bacteria because it is useful in increasing the organism's virulence or ability to cause disease. It is helpful because it prevents our phagocytic white blood cells from ingesting the organism. So it's very useful to the organism as increasing its pathogenicity. It's also useful in biofilms. So if you think about plaque on your teeth, or implants like pacemakers, or knee replacements, hip replacements, capsules help an organism colonize those areas. It allows it to stick to the surface, but then it also enables it to stick to other organisms, and they become this sticky mat, and like you see here, with a variety of organisms, not of the same species, that then colonize that location. So that's the end of 3.2. Check that you understand the external structures and their functions, and I hope this video was helpful.